Welcome back, everybody. Really pleased to see everyone again today. This is our fourth session in the Cerebral Blood Flow virtual seminar series. For those of you who don't know me, I'm Caroline Rickards, one of the co-organisers of this with Dr. Patrice Prasad as well. Today we have Sam Lucas as our keynote speaker and also chair of this session. But just a few rules for the road as we've always gone through so far in these sessions. So just some rules of engagement. Please make sure you keep your microphone on mute and your video off throughout the session. There will be time for questions at the end of each presentation. You can ask questions through the chat feature, and Sam, Pat, or myself will be managing those as we go through. Or you can actually raise your hand and ask the question if you'd like to. The session will be recorded and is already being recorded right now, and then we'll be posting all of the video on the Keynote website in a couple of days, which is also the videos that are loaded onto YouTube. If anyone would like to be a member of the Cerebral Autoregulation Research Network, please let me know. I'm the secretary, so just send me an email with your CV and I'll add you onto our mailing list. So I'm going to hand over now to Pat Prasad, who's going to introduce our first speaker.
So hi everybody, again thank you for your presence here. So it's my great pleasure to present the keynote speaker of today. Dr. Sam Lucas is a senior lecturer and PI at the University of Birmingham in the United Kingdom, and the title of his talk is Exercise and Cerebral Blood Flow, Optimizing the Stimulus for Brain Vascular Adaptation. Really looking forward to it. Sam?
Cool. Firstly, thanks both Carolyn and Pat for organising this. I think yeah, this is a wonderful opportunity for lots of us to be able to present, you know, in a time where we can't leave our own borders. So I really appreciate all the work and the effort that you guys have put into this, and yeah, really excited about how the sessions, the seminar series is going to shape out. So I guess I'm going to present on an area of work that I do. For those of you that don't know me, my name is Sam Lucas. I'm a senior lecturer here at the University of Birmingham, aligned with the School of Sport, Exercise and Rehabilitation Sciences, and part of the Centre for Human Brain Health. So the work that I do really is in exercise and environmental physiology. We've got a research group within our school where we've sort of put together a number of PIs, and I guess collectively we're, I guess, the vascular and environmental physiology research group. And within that group, I guess, amongst lots of things, a key interest for me is exercising brain and how we can improve brain health, as well as how we can measure it, which is another can of worms, I guess. And I won't really speak much of that tonight, but yeah, I guess they're my areas of interest. As a New Zealander, it's important in our culture to sort of tell you a little bit about where I'm from. So as I say, I'm from the University of Birmingham, but hopefully you recognise that I don't sound much like a Brummie. I'm actually from New Zealand, which is over here in the bottom right-hand corner of the globe. And we're famous in New Zealand, of course, for mountains and landscapes and lakes and things. Some of you may know New Zealand quite well through the Lord of the Rings. And of course, we have a very famous rugby team, which is so these are sort of, I guess, our key tourist attractions. And at the moment, we have a very high-profile Prime Minister as well, who's making waves around the world and promoting New Zealand. So that's where I'm from geographically. Academically, you know, these are the two giants, I guess, that I stand on their shoulders. So I did my PhD with Jim, the University of Otago, and then my last study with my PhD. Phil Ainsley arrived and we started working together, and then I went on and did a postdoc within his lab. So I've been incredibly lucky to work with such brilliant people. I guess in this environment, you know, there's a number of people here who are, I guess, commonly known as being part of the Ainsley Army. Well, I guess I'm part of that as well, and also part of the Cotter clan. There's a number of us as well around the world now that have been, I guess, trained and brought up under Jim. So really, so as was sort of been mentioned, you know, our session is really around looking at exercise and how we can use exercise, try and optimize exercise to improve or optimize brain vascular adaptation. So my job really tonight, to start with, is to give us a bit of background here and sort of set the scene for the talks to follow. So Rhodri's going to talk around a study where we've compared running versus cycling. B's going to talk about some cerebral haemodynamics during running. Sam Weaver is going to look at some differences between different exercise protocols that we use. And Leena Shoemaker, who did a PhD with Jim out of New Zealand, and I helped out from afar. And she's going to present one of her studies from the PhD, looking at cerebral blood flow responses during swimming. So hopefully you can kind of see there's a real theme around exercise and how that may influence and change brain blood flow, and that's sort of what we're going to look at tonight. So hopefully just as I guess a background slide, this should be very familiar to everybody really, that blood flow obviously to and within the brain is vital for our survival, and it's important of course because we have limited storage capacity within the brain. Therefore the regulation of blood flow is important, and as a way I guess to reflect its importance, we know that the brain's only 2 to 3% of body mass, yet it demands a large amount of our cardiac output and things. And we know that when we have issues or problems with blood flow, we have events like stroke, and that can be caused, of course, by either over or under perfusion. So there's also functional consequences linked to brain blood flow. It's important for performance, sport performance, cognitive function. As I've mentioned, its impairment, either absolute flow or its regulation, are linked with a number of different diseases like dementia, stroke, traumatic brain injury. And we can use the regulatory capacity or its responsiveness. It has some prognostic value in terms of predicting mortality. And I guess we have this continuum of challenged or impaired brain blood flow regulation. At the acute end, when we have challenged or impaired, we can have events like fainting or stroke, which are, I guess, associated with those sorts of issues. And then through towards the chronic end, more long-term impairment of brain blood flow is associated with a number of diseases, hypertension, dementia, and of course concussion, traumatic brain injury. And some of these we're going to talk about in some of these later seminar series. And for me, as an exercise physiologist, that's my background. That's what I'm passionate about. I'm really interested in knowing what we can do to what's the role of physical activity in improving this, and in a way that I guess the component stressors that are within exercise. How can we use these better or at all to improve brain blood flow and its regulation? Of course, just to, I guess, to place it in the context that we live in and research in, the link between physical activity, aging, and brain health is pretty well established. Of course, we're living in an increasingly aging population, society, and sadly also becoming more sedentary. And both these things have negative implications for brain health. And the good news, of course, is that habitual physical activity or exercise has a positive influence on this and can offset some of these declines. In the context of brain blood flow, here's a nice set of data that was produced, a combined effort, I guess. So Phil Ainsley, when he was at Otago, combined with the group up in Liverpool, John Moores, and pulled together 300 participants, 150 in each of these groups, and showed that while both trained and untrained people, there was this natural age-related decline that we expect. Those that were habitually fitter offset this natural decline, and it was in the order of about 10 years, which is great news. Some more data. Again, Phil's involved here. This time with Damien Bailey out of Wales, showing again in young group and an older group that there's this positive relationship between aerobic fitness and brain blood flow. Here measured with transcranial Doppler, and you can see here, of course, there's the lower fit, the sedentary group have a lower absolute relative fitness versus the trained people, both in the young and the old. And they also looked at cerebrovascular reactivity, the responsiveness to CO2, which is becoming a key marker of brain health. And again, see the same positive relationship here. Sedentary populations, both young and old, had lower reactivity versus their trained ones, and a really strong, pretty good correlation here. Of course, the issue with these data is that they're cross-sectional. So one key question really is, can you, anywhere along this blue line, as it were, right across the age spectrum, can you take that individual and move them up to the red line, to the trained line, and increase their brain blood flow? And one of the first studies that did this was again out of Otago, led by Phil and Jim Cotter, and Carissa Morrell was the PhD student that did this. This was one of the first studies I was involved in as a postdoc with Phil, and we showed that with just 12 weeks of standard aerobic training, circuit gym staff, walks, some runs around Lost Creek in the bush up there, that you could increase brain blood flow both in absolute terms, again measuring using this transcranial Doppler technique, and also showed that the cerebrovascular reactivity was also elevated. So this is great news. Longitudinal studies, of course, are always more powerful, and then cross-sectional and building evidence. It's not the only study. In fact, prior to this, Ivy and colleagues showed that in stroke survivors with a 6-month treadmill aerobics training intervention, you can increase cerebrovascular responsiveness by almost 30% on both sides. And this was associated with an increase in aerobic fitness. And these two changes were pretty well correlated. And this was against a control group where across that same 6-month period, with their decrease in fitness, and some natural declines and some really marked declines, depending on the side of measurement here in cerebrovascular responsiveness. One of the interesting things, I guess, within this data set is when you go in and look at, I guess, the subgroups within this training cohort. This effect was really driven by those that weren't on statin medication. So I guess the message I tend to give the students when I show this is, there's two messages here, that the drugs work, but exercise also works, and puts them back in the same in the same I guess same ballpark as that. So a non-pharmacological option is good. So how does it work? Well, Pat showed this a couple of seminars ago, where we've got a number of different mechanisms, both direct effects from exercise that happen within the brain, where we have increased blood flow moving through the vasculature, which releases a number of different mediators. We've got increased neural activation, of course, which is sending signals, and these things are having a direct effect in changing brain structure and function. But not only direct, I guess, mechanisms, we've got a number of indirect ones. Mediators being released from the systemic tissue and the cells, which are then feeding back or feeding up towards the brain to then drive these changes. And of course, we have these again indirect changes or exercise-induced effects on the cardiovascular system as a whole that has a positive influence on brain health. So we've got direct effects in terms of some of the structural and the functional changes, and these indirect effects where lowering of blood pressure, lowering of arterial stiffness, decreased inflammation, and all these things are positive things for brain health. I think it's always important for us who are maybe perhaps a little bit obsessed with the brain is to remember that we don't lose sight of the fact that there's other tissues and organs apparently which are important to consider as well. So exercise is amazing, has positive effects on the brain, but also is good, of course, for our whole body health. But we know this, right? We know this. It's accessible, it's effective, it's multi-potent, it's safe, and it prevents most of these, I guess, are lifestyle-induced modern chronic diseases. But the problem is, right, that we don't do it. Current recommendations of of 30 minutes most days of the week are not being met. I mean, and the proportion of those not meeting them, depending on the demographic, is really high, especially when you actually measure it objectively. And we know that that inactivity is now a top risk factor for poor health, and strongly associated with cardiovascular mortality. And here in the UK, so these are statistics on physical activity, reported physical activity, self-reported physical activity from last year. And I guess it doesn't look too bad, I guess, in terms of some of these younger populations. It's self-reported, so perhaps if we measured this properly, or objectively rather, these levels might actually be much lower in terms of meeting recommended guidelines. And I guess the big concern here is this decrease, in activity levels as we age. One of the scarier plots within this report that I looked at is when you look at children and young people, and it's frightening in terms of the kids meeting what's expected. Sure, it's an hour, not half an hour, but only 18% across both boys and girls are meeting this guideline. So to me, this almost seems like a ticking time bomb. We've got an increasing older population, which we know exercise less, but we've also got a young people who are not doing much. So where are they going to enter? I guess this bottom line here, it could get pretty dire if we don't do something different soon. So I guess it kind of, I think it's really time for us to rethink what we're doing, and I think that kind of starts with understanding how exercise works, and what types of exercise are best. For example, comparing high intensity interval training versus continuous exercise models, trying to understand how it works through the mechanisms, and through this information, this knowledge, we can improve our exercise prescription guidelines, as well as explore other options that can access the health benefits for both healthy and diseased populations. So one of these models, Pat's talked about this already in this series, so I won't spend too much time on this, but that's, for health, it's been around in the sporting athletic arena for a long time, but it's really sort of taken off in the last 20 years or so around being used for health benefits. And a broad definition, I guess, of high intensity exercise is bursts of exercise. So this can be anything from all out, really short bursts, through to more clinical models, which are high intensity, sort of 85% of heart rate max, but these bouts are a little bit longer, interspersed with recovery, active or passive rest of recovery. But yeah, I guess that one of the keys, I guess, selling points for these is that they take less time, and that's a common barrier that people put forward for not engaging in physical activity. And I think it's difficult to mention high intensity exercise for health and not mention someone like Martin Gabriella. Marty's done some great work in this area over a long time. And here's a recent review that was published at the start of the year. And you can see he's really sort of come a long way, and some of the more current models he's been looking at, things like stair climbing, 1 minute bouts, and seeing the efficacy of real world HIIT exercise paradigms. And his focus is really around cardiorespiratory fitness, metabolic adaptations, and there's some really strong evidence from his work, from lots of groups around the world that really illustrate the power and the effectiveness of this type of exercise. And for some markers, it's over and above what you get with what's traditionally recommended. But what about the brain? We're still in a scenario where there's still no randomized control trial that's actually focused on brain blood flow responses. And in fact, there's very few that have looked at any brain function outcome measure. So there's still lots of work to do in this space, as Pat highlighted in his series. So I kind of just want to go back to this, I guess, this question about how does exercise work? and I guess it's to come back, and this is something that Jim instilled in me really, was around understanding exercise from a first principles approach. So, you know, it's actually the strain of exercise, of course, which is driving the adaptation. So, you know, a number of different types of exercise, or I guess the parameters within exercise will induce a variable strain, and that's what's going to elicit this adaptation. Kind of simply speaking, you can give the same person the same stress, but that's going to have a markedly different physiological strain between these two individuals, for example. You know, and these two individuals will have a markedly different adaptive response because the strain of the same stress was different. Okay, so we can kind of expand this out. There's a number of physiological strain mediators that are driving the responses and ultimately causing the adaptive response. We can play around with the strain response by changing the exercise parameters. We can also introduce environmental factors or nutritional factors, and then there's going to be some modulators which also alter this strain response, ultimately changing the muscle functionality, power, strength, of course changing, affecting, adapting the cardiovascular system, and then direct effects on the brain structure and function. So as Jim is highlighting here, you can do a lot just by changing some of the parameters around exercise, and there's lots to learn in this space, and this is what our work here in Birmingham, we're doing a lot of work around this space, and we'll present some of that information, some of those findings tonight across our talks. And I'm going to focus a little bit on some of the water immersion work that we've done as well. So Claire Burley did a PhD with me a few years ago now. She's now headed off to Australia, to sunnier shores. But she, I guess, reimagined some of these, this idea that I just presented before, and kind of putting the brain cognitive health right at the centre. You can sort of see the different types of exercise or intervention strategies that we can apply, which will induce a physiological strain that will result in the adaptive mediators and ultimately affecting and changing brain health here. So this is, I guess, a focus on the brain, and we can do these different exercises either singly or in combination, and trying to, I guess, find what the optimal one is, I guess, for us, or is an important question that we're after. So as I say, I'm going to focus quite a lot around physical activity, and we've got 3 talks really that are looking at the differences in the exercise modality, the effect that has on particularly brain blood flow, cycling versus running, some of the oscillations that we get with that. Leena's going to talk about some swimming responses, and Sam's going to look at what happens when we change both the intensity and the duration, and look at some common exercise strategies or protocols that we promote. And Dr. Blake Perry is going to, I think in a later seminar series, is going to talk to this as well. So I think we're going to really, across the series, do a really nice job of covering what's going on across a number of different exercise modalities, and changes in exercise intensity. Before we kind of jump into some of the data, just to sort of rewind a little bit and just remind ourselves, I guess, of how the regulation of brain blood flow works. So this is taken from a nice review by Kurt Smith and Phil Ainsley, and showing these key regulators, factors that influence brain blood flow, and of course the metabolism, neural activation, blood pressure, sympathetics. I can leave that one for Pat to talk to, but these are this is, I guess, the common trend that we're looking at. Now, of course, the chemical stimulus is really important, particularly CO2, and we'll talk a bit about that. And cardiac output as well has a direct effect on brain blood flow. So during exercise, well, in any context, this is complex. It's multifactorial, and they all work together, and when you throw exercise in there, exercise affects each of these different factors, and it also changes their interaction. So it's a complicated story, and I'm particularly interested in looking at how exercise, the different types of exercise, the different exercise intensities affect ultimately brain blood flow, and that's going to happen through these changes in these regulatory factors. So just to talk about what we think we know, or what we commonly report in the literature around the relation between cerebral blood flow and exercise intensity. So this is one of the earlier studies that reported this pattern. So as we increase exercise intensity, brain blood flow will rise, tending to plateau at around moderate intensity exercise before returning towards rest, at these higher intensities. And it maps pretty closely to these changes in end-tidal CO2. And here's a nice review, again, the same review by Kurt and Phil, who pulled together data from 17 studies. I think at the time that was all the studies in the literature that had reported changes in brain blood flow across different exercise intensities. And you can really, I guess, see this pattern of brain blood flow increasing from rest up and peaking around this moderate intensity exercise before declining back towards resting values, so I guess 0 here, and this is in line with these changes in end-tidal CO2 represented by these red numbers. So this is when we talk about changes in exercise intensity. This is the pattern that we'll commonly report and we'll mention. The question I have really for this is, is this the same for all exercise modalities? All these studies here are from cycling-based work. Quickly scanning that, I think that's correct. But yeah, most of the work that we've done so far has been done on the bike, and that's primarily because it's much easier to measure brain blood flow when people are sitting still. But there are some studies out there which have measured brain blood flow during different types of exercise. So Frank Pott, Copenhagen, of course, they can do anything, and they did this study measuring arterial blood pressure beat to beat, and also measuring brain blood flow with transcranial Doppler. And what you can see here, so these dotted lines are the catch phase of the stroke, and you can see with every single stroke you're getting these increases in blood pressure. It's almost a Valsalva maneuver for each stroke, and that's reflected in the brain blood flow response. We followed this study a few years later. So 5 years ago, Liv Fall was doing her honours degree with Jim and I at Otago, very, very strong honours student. And she was very keen to look at this relationship. So we also measured brain blood flow during rowing, and see the same pattern, I guess, stroke to stroke, seeing this increase and decrease in line with each stroke. And what we did was to look at changes across the exercise intensity. So we measured effectively 4 different intensities. We did standardized protocols, 50 and 75% of heart rate range, and then they did an all-out 2K erg, and then a 30-second sprint at the end. And what we see is, I guess, what we'd expect in terms of our end-tidal CO2 response. When they did higher exercise intensity, end-tidal CO2 went down, but this wasn't reflected in our brain blood flow responses. So the black and the grey line reflect 2 different times of day. So Liv was interested in the time of day effect from a performance perspective, and her rowers doing these 2K ergs, and looking at that, but also measuring cerebral haemodynamics. But yeah, regardless of the time of the day, we see brain blood flow unchanged. If anything, it went up a little bit when we did an all-out sprint, where end-tidal CO2 was going down. So there seemed to be this uncoupling. So the relationship between end-tidal CO2 and brain blood flow seemed different for rowing relative to cycling. Another study, another group out of Copenhagen, I think Lingera and colleagues, measured brain blood flow during running. and they showed, they measured 3 different exercise intensities, not quite all the way to max, but what they reported was this, I guess, this oscillation within the blood flow profile, which is quite interesting. That in terms of the mean value, they do see, you know, an increase, and it wasn't significantly lower than 65, but it was starting to trend down in terms of the mean blood flow. So I guess the issue with this study is that they didn't take it all the way to near maximum. So certainly beat to beat differences in running versus cycling. But yeah, again, running looks like it does something different. And we started doing a little bit of work around running and measuring brain blood flow, linked with a water treadmill study, which I'll talk about later. But during this, what we noticed was this oscillating profile. You can sort of see here, so this is our Doppler traces that we're getting here. And then we use a lab chart software. So I've sort of exported this here, and you can sort of see the beat to beat blood flow trace here at rest, moving into exercise. I mean, you can see this oscillation wavelength starting and then changing. So we have these different frequencies and oscillation durations as they start exercising. So this 1 minute block is the same, and so you can kind of see that this pattern is quite variable. And this is something that Gabriella Imi is going to talk to us about in the second talk. And she became really interested in this and did some neat work around trying to, I guess, manipulate this through different things, and she'll talk to us about that. So, you know, the question, you know, is this relationship of increasing to 65 and decreasing near maximum, is this the same for all modalities? Well, maybe not. It does look like it's different between different exercise modes. However, very few studies have actually formally compared different exercise modalities or look at the effect of brain blood flow across different exercise protocols like HIIT or moderate intensity exercise. So that's sort of the work we've been doing over the last couple of years. Again, Rhodri's going to present all of this data, but I'm just going to give you a little snapshot. So as part of this, we did an incremental exercise protocol where we're doing 3-minute stages all the way up to 95%. That was our near maximal. So we hit 35, 50, 65, 80, and 95. And measure brain blood flow beat to beat during that time. And as we'd expect, as we saw again with running, we see this oscillating pattern beat to beat compared to cycling. So we, I guess, can formally confirm within the same study and the same cohort that the running does elicit this different response. And something interesting that we saw is that when you look at the mean responses, so you average the last 30 seconds of each of these stages, what we saw was both modality and fitness specific profiles for blood flow responses. And so if you can look down here, you can see, so this is our running response. You can see that, or let's start with cycling. You can see that as expected, it increases to 65%, and then decreases as we increase our exercise intensity. Whereas for running, we get something different in terms of our higher fit participants. So we split them into fitness groups, and what we saw is that those with a higher fitness just increased linearly all the way up as we increased exercise intensity. So whereas our lower fit participants, or the participants with lower fitness, their brain blood flow was more like the cycling based one, and this was certainly for the fitter participants, was in spite of the expected inverted U response that we saw with end-tidal CO2. And again, you know, we see this with cycling as you'd expect. So Rod's going to talk more about the study and also present some of the HIIT data that we did with this as well. I haven't stolen all of this thunder just yet. Again, Sam's going to talk more about this, but just to give you a flavour of the work we've done in this space as well. So he was interested in looking at the different exercise protocols. So this is the first study in his PhD where he's examined the acute cerebral haemodynamic responses to these different exercise protocols, and the resulting release of the neurotrophic factors from these. So just to sort of highlight and show you what we're seeing with, or what we saw with the brain blood flow responses. So here, so these are our resting values. So we've got absolute values of MCAV here, and you can see, as you'd expect with our traditional model, which is 30 minutes for exercise of moderate intensity exercise, it increases relative to rest, and it stays pretty constant across that whole period. What we see with our interval exercise is something quite different. You know, it increases during exercise, it decreases with the passive or active recovery, and you can see across the intervals there's the different patterns starting to emerge within this protocol. And then with us all with our 30 seconds super maximal sprint, what we see is quite a marked difference, not only between or compared to the other protocols, but also within this protocol, such that the first interval was quite different to the remaining ones. So Sam's going to talk more about this. You know, I guess with the added data around, you know, what does these different profiles mean for the release of these factors, which are linked to changes in cerebrovascular health. So stay tuned for that study, that presentation. But just to kind of put you in I guess a wider context for what we're doing in our group, is we're really interested in, I guess, in observing these different profiles, both across exercise mode and these different protocols, which are mixing up intensity and the duration. And from this, we're also taking a lot of blood, I guess, during these. So we're trying to understand what the molecular and cellular signaling that's going on. And from that, what we're trying to do is get a better understanding of what's happening, which one of these is perhaps best, which combination of these might be best, so that we can then design and apply this as a targeted intervention to understand how we can use exercise better and measure brain health outcomes. And what we want to do is do this right across, I guess, the broad range of outcome measures. And you know, it's important, I guess, to acknowledge that this is certainly not just me. This happens with lots of different people and lots of expertise across campus. And Sam's doing a wonderful job of keeping all these different people happy throughout his PhD. So I've just talked, I guess, a lot around physical activity, the different modes and different modalities, intensities. And one of the key factors linking exercise-induced changes, particularly in brain vascular health, is the effect of the movement of blood through vessels known as shear stress. So for the last little bit of my talk, I guess I want to focus on that aspect and jump into some work that we've done using water. So as I mentioned, the mechanical movement of blood through the vessels, you know, has been identified as a key exercise-induced mechanism that facilitates improved vascular function. Now, and this is seemingly, you know, primarily through the increase in nitric oxide bioavailability. Danny Green and others have done a really nice review on this, and recently, you know, highlighting the importance of shear stress in terms of improving vascular health. But you know, how can we target this? You know, and especially for those that can't exercise sufficiently to produce an optimal strain adaptive response. That's kind of, I guess, what's got me into working in water. So we know with water, water immersion elicits, you know, a hydrostatic stress. This increase in pressure increases central blood volume, which improves our cardiac performance and increases tissue perfusion. And as a result, these types of water-based activities have great potential to enhance shear stress through the vasculature. And Danny, of course, has done some work in this space. I guess leading the space, should I say? Sorry, Danny. And one of the studies, one of those earlier studies that he did in here was looking at how exercising in water can augment this brain blood flow response. So this is a neat little study where they just did some box stepping for 20 minutes at a pretty low intensity, less than 100 beats per minute, and they matched that with doing the same exercise with no water, and when we see this augmented brain blood flow response. So around that time, I got in contact, or rather Devinder actually just emailed the school and asked whether anyone was interested in working with his physio clinic, because he had a water treadmill, and he thought people might be interested in that. And of course, water treadmills are not new. They've been around for a long time, are used a lot for rehabilitation from injuries, sporting, athletic injury, but also spinal cord injured and gait re-education in stroke patients. And it's through this, the water and the buoyancy unloads some of the mechanics and increases some of the resistance. So there's some really helpful, beneficial, acute and chronic physiological adaptations that can occur within this, within using this modality. So I was really keen, based on the work that Danny had just published, that we could maybe jump in and use this. And for me, one of the key advantages of it was the ability to change exercise intensity, and of course, not having to step up on a box. It seemed perhaps a little bit more real world, for example. So at that time, a master's student who was keen to do some work, so Rhodri kindly led this study for us, and the aim of the study really was just to compare the cerebral blood flow responses and the heart rate responses during an incremental exercise test during water versus running, and then we played around with water levels as well to look what happened there. So our protocol was an incremental test, starting with walking, where we did 2-minute stages up to the maximum capacity of the treadmill, which was 10 kilometers an hour. And then on that point on the water treadmill, we did some different depths to look at the responses with that. Whereas in the land treadmill, we then ramped them up. If they hadn't already reached max, as these 3 individuals did, we then took them to max by increasing the gradient. So what we did really statistically is we were interested in comparing these matched increments. And what we saw was that across that 10 minutes, those 5 stages, brain blood flow in the water peaked sooner and was elevated consistently higher versus our land treadmill exercise. So it peaked here at the second stage, at the 4-minute stage, whereas the land treadmill took all the way to that final 10-minute block. And the difference here, this was an increase of 21%. This was an increase of 12%. So not only earlier, but markedly higher. And this was for a lower heart rate response across that 10-minute block. I also wanted to, of course, because they started running, at the intensity that they were walking at on land. So I was quite interested to say, okay, well, let's compare like for like. So we took the responses when they were walking, and then we used heart rate to measure heart rate to estimate 60% VO2 max, which of course is the recommended guideline. So interested in comparing the brain blood flow responses at these two specific exercise intensities. And what we saw, regardless of whether they were walking or running at 65% VO2 max, we saw an elevation in brain blood flow. And what was really striking for me was that when you compared walking in water to running on land, the brain blood flow was the same. So we were, excited the fact that, perhaps, using water, it makes attaining what we talk about as being an optimal exercise intensity for stimulating blood flow and therefore CS stress. We might be able to access this for populations which, have impaired or reduced physical capacities, and therefore running is maybe not an option for them. As I mentioned, we also played around with water depth, and what we saw is that when we increased the water depth, we were able to lower heart rate, so reduce the heart rate, but at the same time, we were able to we managed to keep, or brain blood flow was consistent across that. So again, potentially useful in terms of reducing the heart rate load, cardiac stress, yet inducing a similar increase in brain blood flow and therefore cerebrovascular adaptation. So from the study, we were able to conclude that aquatic treadmill exercise augmented brain blood flow across a range of exercise intensities and for a relatively lower heart rate response. And it was particularly evident at lower exercise intensities when they were walking and running. And of course, this elevated flow has a potential to enhance shear stress and ultimately drive changes and improvements in brain health. And it appears like it's an ideal modality for those with reduced physical capacity, such as stroke survivors, where their reduced capacity may limit the exercise-induced physiological strain necessary for stimulating adaptation. So we took this idea and in a pilot study, another master's student, they sign up for a 6-week project with me, and they end up doing a 4-week training intervention with stroke survivors. But Karen was brilliant, supported by Claire Burley. This wasn't part of her PhD, but she helped out on the study. So we took 6 chronic stroke survivors, all 2 years post their event, and it was a range of ages from mid-30s into mid-50s. And the key measures that we took here were looking at brain blood flow responsiveness, as we've talked about. That outcome measure is linked with stroke risk and links to brain function. And we also took some functional measures in this population, which is really important. So of course we did our baseline measures. We then exercised them for only 4 weeks, 3 sessions a week, and pretty low intensity, 20 to 30 minutes, just walking in the water, and then we took them measures at the end of this period. And the findings were really striking. In terms of our cerebrovascular responsiveness, if we look at the increases that we saw in both the affected and the non-affected hemisphere, these increases are pretty big, 40%, 65%. And if you put that in the context of the other study that's worked with stroke survivors and measured cerebrovascular activity, it's nearly twice like what Ivy and colleagues show. So as well as the vascular ones, we saw some really impressive improvements in gait function, and not for everybody, which is interesting in itself, but for 2 people of the 6, their 10-meter walk speed increased. That should be 1.5, 1.5 meters per second, which is well above the clinical, the threshold for clinically meaningful change. And perhaps what's even more important is that they really loved it. The participants wanted more. It was well tolerated, and we didn't lose anyone out of the study. So we were really encouraged by this. So at the moment we're trying to sort of take this forward and get funding, and that's, as we all know, it can be challenging. I just sort of wanted to, I guess, highlight some work from Danny Green and his group. Again, as I mentioned right at the start of this water treadmill, this water-based stuff, they've kind of been leading this space. And Danny's just starting to publish work with his group now on this Preventia RCT trial. And the overarching theme or question they have is, again, using water to enhance the arterial shear stress, and they're primarily interested in cerebrovascular function and cognition in an older population. So they've recruited 70-odd participants, but I think in most of the studies published to date, there's about 60 from that. They're all older, healthy individuals, and they've done some land walking and water walking, and they've also got a control group. They've done 24 weeks, 3 visits, 50 minutes each visit. And just to kind of give you a little snapshot here of some of the results I've published so far, they're seeing similar improvements for land and water and VO2, Interestingly, the FMD, so their peripheral vascular responses, is better on land than is in water. It's a bit of a shame. Yes, typical for the Aussies to kind of spoil the story for us. But still haven't published. I know these data are coming, and Danny was going to be presenting this at ECSS in a symposium that he, Kate Thomas, and I were going to do. So I know that they're around somewhere. So looking forward to seeing these data. But this is a really neat study, and certainly showing some positive effects. There's also some body composition findings which have been published, and they're showing that water might be better for that. So I think the take-home message out of this is that it depends on your outcome that you're interested in, and water might be useful in some contexts for sure. This is a review that was published just last month, June, talking about the potential. So there's lots of interest in this space using water-based therapy for that, targeting neurorehabilitation. So I think there's lots of scope for that. I'm working pretty hard to try and get something like this here at Birmingham. Thought I was getting pretty close. However, I think that the pandemic has sort of cancelled a lot of our capital work. So I think in the university, so I think this is on hold. But working with a partner here, Malcolm Davis at DWS, who do these pretty cool water treadmills, either in the floor with bikes as well as another option, or these drop-in treadmills into a current pool. So lots of work to be done, and we're trying to push forward with this. So I kind of talked about physical activity and the different options we have there. We've talked about hydrostatic stress. Thermal stress is another great option, and Jim has been leading a lot of this work, and I'm sure these guys, Kate, Luke, and Ashley, have been doing work in this space, and maybe presenting some of this in their seminar later in the year. Hypoxia is another option that we can use potentially to derive some of these physiological strains. Nutrition, Katerina Rendera is a colleague here at Birmingham. She's done some really neat work, and I think we should probably have I'm nominating her to run a session on nutritional nutraceuticals and brain health. I think that would be really cool, and she's got some really neat data that support that. And of course, cognitive activity. There's some neat work that's coming out of Australia that's combining cognitive loading and exercise, and looking at whether that augments some of the exercise-induced changes in brain health. So just to finish, I guess the questions that I guess drive a lot of my work is trying to understand what it is about exercise that improves brain function. What are the mechanisms for adaptation? And can we get this from individual stresses, either singly or in combination? Can we combine that with exercise, for example? And how best to target these mechanisms? Looking at the different parameters of exercise. Is a one-size-fits-all for whole body health? Is that really appropriate? Perhaps we need to think about targeted interventions for brain health, targeted for the brain, targeted for the individual, with specific population, and of course real life. I think it's really important that whatever we're designing, we're thinking about its applicability, its validity out there in the real world. Just to finish, a lot of people to thank. I guess you can't do any of this work really without support from your postgraduate students, and I've been very lucky to have a great team. So these are the students that have supported the work that I've presented here, collaborators here at the university, within our wider research group, and that of exercise metabolism within the school, and physiotherapists that have helped with me, and then across campus from different institutes and centres, supporting the work that I'm doing, and of course colleagues from across the globe, and a special shout out to our New Zealand colleagues, and Pat, Damien, Phil, and Tom have been working in this space as well, and then of course funders and support partners that have helped help produce this work. And thank you for your attention, and I welcome any questions.
So thank you, Sam. We may have time for one or two quick questions. So one from Sandra Bellinger. So do you know where the stroke lesions were and what acute treatment the stroke group received, TPA versus stent removal?
You know, we didn't ask that question, which is a real shame, and I wish I had. So yeah, we didn't ask that. We didn't have ethics at the time. I hadn't thought about that, but I have been asked that question a few times. So yeah, we are certainly wanting to get that information going forward. But yeah, good question.
Next question from Ron Panery. Can we ignore the contribution of mean blood pressure in the interpretation of changes in MCA velocity during exercise?
No, I don't think so. Pat might want to answer this question. No, I think that certainly for when we're looking at cycling, I think CO2 is potentially more important. Certainly, well, when they're actually exercising, I think what's interesting is when we stop exercising, the effect of blood pressure seems to be apparent, maybe more. You know, and again, Pat's been doing some work in this space, and we've seen a similar thing when you stop a high intensity interval bout. There seems to be this rebound that happens, and that might be linked with blood pressure. So I think it'd be very, very hasty to say that it's not important. But there does seem to be in some modalities at some intensity that CO2 is maybe more influential. But say, for example, with rowing, you're certainly seeing, and running, you're seeing these beat-to-beat oscillations which have been driven by blood pressure. So blood pressure is definitely having an effect. Yeah.
So maybe one last question from Rian Junerio. The great talk, Sam. Quick question. What stressors do you think are driving the different divergent responses between FMD and brain blood flow and cerebral CO2 reactivity when we are looking at water versus land walking?
That's definitely a question for Danny Green. I don't know. It's interesting. Again, it would be nice to be able to see the brain blood flow data in that context. But yeah, I guess the limbs are in the water, so maybe the hydrostatic stress is suppressing some of that shear stress. I'm not sure. You know, and it might be different for what's happening in the head. Again, I think Danny's probably better to answer that question, but I guess it would very much depend on whether they see an improvement or a greater increase in their brain responses versus their FMD. Because if they don't, then you know, I guess we maybe have to rethink what we're doing, or maybe that for that population, healthy older population, you know, the benefit isn't as great. But yeah, again, I think we need to probably wait until we see those data. But it is interesting that it didn't seem to, I guess, magnify that effect in the peripheral vasculature.
Great, thank you. So we have a lot more questions in the chat room. So if you want, you can answer that as we go on. So we'll move to the next speaker. You want to introduce it, Sam?
Sure, no worries. So Rhodri Furlong is a master's student, or was a master's student in my lab. He's very kindly taken time out of his working career these days. He's working as a vascular scientist down in London, which is why we're doing it quite late, trying to fit around his work timetable. But he's going to present some work around, as I presented earlier, looking at the differences in exercise modalities across an exercise intensity range. So over to you, Rod.
Thank you, Sam. Just get the slides up. Right, so good morning, afternoon, or evening, depending on where you are listening from. As Sam explains, we're going to be looking at the effects exercise modality and intensity have on cerebral blood flow. So to kick things off, we're going to look at what we know, which is that exercise-induced elevations in cerebral blood flow are associated with positive cerebrovascular adaptations, likely via the stimulation of shear stress and cyclic strain, and the upregulation of nitric oxide and neurotrophic factors such as VEGF, brain-derived neurotrophic factor, and insulin growth-like factor 1. So what we know about cycling during cycling in cerebral blood flow is that there is this biphasic relationship where cerebral blood flow increases up to moderate intensity exercise and then decreases as exercise intensity is elevated, and this is mirrored by the end-tidal carbon dioxide profile as shown on the right of your screen for active and sedentary participants. Less is known about the impact that running has on cerebral blood flow. The paper that Sam also discussed by Lingira suggested that there might be an inverted U relationship between exercise intensity and cerebral blood flow velocity profile, but it was non-significant. But interestingly, they did show that there were oscillations in the cerebral beat-to-beat blood flow profile that had originally been shown in the peripheral vasculature by Palatini and colleagues 2 decades beforehand. What we don't know is the optimal exercise modality and intensity to maximize increases in cerebral blood flow and the associated pathways. And therefore, the aim of this study was to gain a better understanding of the cerebral haemodynamic responses to running and cycling at a variety of exercise intensities. So 26 participants took part with a mean age of 26. And our primary measures were middle cerebral artery velocity via transcranial Doppler ultrasound and end-tidal carbon dioxide. So when participants first arrived, they completed a graded exercise test on the bike or treadmill, sort of in the first two visits, and these were randomly allocated. And then on their third and fourth visit, they completed the exercise session, which included exercising at 35, 50, 65, 80, and 95% of their VO2 max for 3 minutes. And these intensities were predetermined via the graded exercise test they completed earlier on. So we found that as expected during cycling, middle cerebral artery velocity and end-tidal CO2 shared this inverted U profile, which is consistent with the literature. But during running, middle cerebral artery velocity increased linearly with exercise intensity, despite a biphasic end-tidal CO2 relationship. And now, interestingly, when we were analysing the data, we noticed that participants who were typically fitter showed the increase in cerebral blood flow with exercise intensity, whereas participants with lower fitness levels didn't. So we ran some stats and showed that for the fit group, which was defined as having a VO2 max over 45, that middle cerebral artery velocity increased linearly, whereas the unfit group, it decreased, and there were no differences between fitness groups for end-tidal CO2 or middle cerebral artery velocity. As previously reported, and as Sam also alluded to, oscillations were present during our research during running across all exercise intensities, regardless of fitness level. But this will be sort of touched upon in greater detail in a later talk. So the sort of take-home messages so far would be that cycling increased middle cerebral artery velocity and end tidal CO2 up to 65% as expected, and then it returned to baseline levels, or towards baseline levels, should I say. Whereas during running, middle cerebral artery continually increased up to 95% VO2 max, and subgroup analysis indicated that this was seen in fitter individuals and not those with lower fitness levels, and also that there were modality-specific differences in beat-to-beat cerebral blood flow patterns. So far we've just discussed cerebral blood flow and sort of a ramp incremental protocol, but due to HIIT's emerging popularity within clinical and non-clinical populations, specifically Western's clinical HIIT model, which I know was discussed in a previous seminar with the 4x4 model. We don't know the effect that this has on cerebral blood flow compared to steady state exercise. So we had a look at this as well and compared the profiles of clinical HIIT against those of moderate intensity exercise for running and cycling. So there were 14 participants in this study. They completed the sort of ramp protocol, and we just focused on the data from 65%, and then compared that to the data of the clinical HIIT model. And we found that the first HIIT bout induced a significantly greater elevation in middle cerebral artery velocity compared to exercise at 65% VO2 max. And a possible take-home message from that is that a single 4-minute HIIT bout of running or cycling may be more beneficial in stimulating cerebrovascular adaptations compared to moderate intensity training. Thank you for listening, and if you've got any questions, feel free.
Nice one. Thanks, Rod. Perhaps while we're waiting, if you've got any questions, perhaps just sort of write them in the chat, and I'll read them out. So perhaps what were some of the challenges that you faced with this study? I guess people haven't measured a lot of running brain blood flow. Did you find that difficult? And I see in the picture there you had an arm pressure, blood pressure cuff on there. So did you try to measure blood pressure?
Yeah, so blood pressure was tried to be measured, but we found, especially during running, that the sort of heel strike disrupted the signal. So that wasn't possible during running, and occasionally we had issues with the TCD probes on the side of the head. But we did manage to get a good data set eventually.
Cool. And what's next? Where do you think this work should go?
So potentially going forward, it'd be good to apply these models, especially the clinical HIIT model, within a clinical population. So sort of similar to the work that you were describing before with the treadmill stuff. It would be good to get access to some patients, be that stroke survivors or patients with vascular disease of some nature, and see how they respond to the clinical HIIT versus moderate intensity exercise.
Cool. So we've got a question here. So this is from Phil Ainsley. Great talk. What do you think about the issue of end tidal CO2 overestimating arterial CO2, particularly in the context of these different types of exercises?
So something that we know they're similar at rest, but obviously during exercise, it does it doesn't match up perfectly. I think we figured that, especially during HIT, if it was it would underestimate the effect that we'd seen, essentially.
And do you think that the different exercises, so running versus cycling, do you think that issue will be any different?
Off the top of my head, I'm not entirely sure. I think I'd probably have to have another look at the data set.
Yeah, I mean, we know that there is some data out there that talks about different vibratory responses, right, between running and cycling. So maybe that might mean that there's different, that relationship between end-tidal CO2 and arterial content could be different just because of the breathing pattern is different. So yeah, potentially, it's certainly something to, I guess, to figure out. But yeah, in the context of looking at how blood flow changes with the different intensities and different modes, how it's regulated is certainly interesting. But in terms of the consequences of shear stress and driving vascular adaptation, yeah, it depends on your slant. Are we interested in how it's regulated, or ultimately the outcome of changes in flow? Yeah. A couple of questions. There's one more question maybe, and maybe Rhodri, you can answer these within the chat. So why did we choose not to prescribe exercise intensities through the ventilatory thresholds? In terms of looking for moderate, heavy, or severe, why did you choose that these different exercise intensity, targets?
Yeah, I guess we went on instead of VO2 max. Yeah, we went off VO2 max simply because that's what I'd seen in the literature, and tried to match it to what had been done before, and obviously put our own spin on it. Yeah, that's why that was done. Yeah.
Cool. Yeah. Okay. Thanks, Rhodri. We'll keep moving. But yeah, great talk. Thank you.
Thank you.
So our next speaker is Gabriella Imi, and she was involved in the study as well that Rhodri's just presented. And one of the things that really took her interest was looking at these blood flow oscillations that we're seeing with running. So Bea's going to talk a little bit about this, and she then took the study on in her master's project to kind of explore this peak phenomenon. So thanks, Bea. Over to you.
Thank you, Sam. So as Sam and Rhodri both said, we know that exercise increases brain blood flow and this shear stress stimulus, which induces improvements in brain health. So currently literature is saying that 65% VO2 max is optimal for brain health due to eliciting peak increase in cerebral blood flow. However, this is predominantly done in cycling studies. But what I'm really interested in is that it's seen in running induces this beat phenomenon, which was first really early explored in Palatini's research, which is shown up here, during track running outside and in the peripheral vasculature. And this was due to a summation of two pressure waves of near equal frequency. And so one was this mechanical wave, which was induced by the step-to-step frequency of running. And the second one was this arterial pulse pressure wave, which generated from the heartbeat. And then later work by Lingira, so he had individuals running on a treadmill at a variety of intensities, saw this beat phenomenon in the peripheral vasculature, and it transcended into cerebral blood flow. And it was in relation to these stepping frequency of running. And he also noted that when an individual was running, they lost their running rhythm when there was a change in the belt speed due to increasing intensity. And this also correlated with a loss in this oscillatory pattern. However, once they regained their running rhythm, this oscillatory pattern came back. So that's just directly shown there's a direct relationship between this step frequency and this oscillatory pattern. What was also really interesting, I thought, that there was an intensity component. So we're looking down here at this trace. So at 50% heart rate reserve, you can make out around about 7 full oscillations there. And then if you then compare this to 75% here, you can only really see one full oscillation or 1.5. So there's a much larger wavelength here, although it wasn't really explored why. Also, Lingera didn't measure PCO2, which we know is a key regulator for cerebral blood flow, and it didn't bring these individuals near intensity, near maximal intensity. So there's still lots more to explore. So really briefly touching on the data that Rhodri presented here, he showed that in these high fit individuals, their cerebral blood flow increased with intensity, so peaking at 95% here, which was significantly higher than during cycling at this intensity. So this is just showing that running is doing something unique in these highly fit individuals at these high intensities. But bringing it all together now, I've just made a quick diagram. There are so many potential factors affecting this oscillation profile. But what I'm really interested in that hasn't really been explored is how does manipulating this heel strike during running, as well as the step frequency, impact these pulse pressure oscillations, as well as what does this mean in terms of shear stress and thus long-term brain health and these vascular adaptations. So what I really want to focus on is how changing this heel strike force, as well as step frequency, how does it impact cerebral blood flow and these pulse pressure oscillations, as well as looking at cycling, more acting as a control. So what I did, there were 3 main protocols. So first I did gradient running. The running biomechanics data has shown that as you run uphill, it initiates more of a toe-weighted run, whereas when you run downhill, it's more hill-weighted. So I also use a heel strike force and step frequency protocol, as well as a steady state cycle. So my main measures were looking at middle cerebral artery velocity using a TCD, as well as some respiratory variables. I'm just going to go into more detail now about the protocols, starting with gradient running, which started with a flat 10-minute run at 1% gradient, followed by either uphill run with an initial gradient of 6% or a downhill run with an initial gradient of -6. So then these gradients and the speed of the treadmill was manipulated so it could reach an individual 65% VO2 peak. Moving on to the step frequency and foot strike running protocol, this consisted of 5 5-minute bouts. So we started with a flat run, followed by a toe-weighted and heel-weighted run. This was just to really target, isolate, and change the nature of the heel-toe running biomechanics. And it followed with a steady state audio cue, so individuals ran to a beat of 145 beats per minute, followed by a random audio cue. So the audio cue rhythm changed every 30 seconds, and it was somewhere between 145 to 185 beats per minute. And then finally, we just finished off with a half-an-hour steady state cycle. But before I go into my results, I'm just going to explain how I quantify these oscillations. So we looked at oscillating frequency, which was counted across a 30-second period at 65% VO2 peak for gradient running and cycling, and then for the final 30 seconds of each running style. So as you can see by the orange brackets here, we just counted these oscillations. And then we also looked at oscillation duration, such as the wavelength, which was average of these 3 last oscillations of these 30-second periods. So you can see that by the 3 blue arrows. So moving on to results now. So this is an individual's MCAV trace during gradient running. And just by looking at this blue trace here, you can see that this oscillatory pattern is not uniform within a condition as well as between. So you can see it changes. So looking more in detail now, these 30-second traces in orange, you can see during flat running, there are 12 full oscillations there. In comparison to downhill running, where there's only 2, so there's a much greater wavelength here of these oscillations. And down looking at uphill, you can see about 2.5 or 2 full oscillations. So this clearly shows that as this individual was running at different gradients, this induced a different oscillatory pattern. However, though, looking at an overall group effect, there was no significant change in this oscillatory wavelength or frequency between these gradients. Although if you look quite closely at this graph, there are some on-average differences. I believe this was due to there being such a large variation in this oscillatory pattern between these individuals, as well as there only being a really small sample size, so therefore there's a power issue. But definitely more research is needed to explore this further, as well as a much larger cohort. So swiftly moving on to heel strike force now. So again, this is an individual's trace. And just by looking at the blue trace here, you can see there is a difference in this oscillatory pattern, particularly if you look at toe-weighted, where they're very narrow, to heel-weighted, where there's much wider and less of these oscillations. So in these traces at the bottom here, it just shows the last 15 seconds of flat running, where you can see there are 4 full oscillations. And as soon as this individual initiated this toe-weighted running, the amount of oscillations doubled to 8 and a lot more narrow. And then just looking at the last 15 seconds of toe-weighted running, you can see there are 7 full oscillations. And again, as soon as this individual initiated heel-weighted running, it drops right down to 2 and that wavelength increases. So just by looking at these traces, you can see that as you target the heel strike force, there is a change in this oscillatory pattern. So something's going on there. However, looking at the group effect now, there was no significant change in this oscillation wavelength or frequency. Finally, looking at step frequency, just by looking at these traces here, you can see none of these patterns are the same. And there was such a large variation in this that overall we didn't find any change or statistical change in the oscillation wavelength or frequency. So as I said, we also use cycling more as a control, and as we expected, no oscillations were present during cycling across all participants. So you can just see here in this blue trace during cycling, they're just non-existent in comparison to the trace above. So this is just confirming that running and cycling induce these different beat-to-beat profiles. Lastly, we actually had an incidental finding of a fitness effect. So when participants first came to the lab, we completed a VO2 peak test. This was mainly just to check for a good signal window using a TCD, as well as getting the individuals used to wearing the headset whilst running. And what we found just confirmed what Rhodri presented, was that in these high fit individuals, when we compared stage 1 of the fitness test to the final stage of the test, MCAV increased in these highly fit individuals, yet regardless of fitness level, end-tidal CO2 decreased between these stages. So it just shows that something's going on here in these highly fit individuals at this high intensity. But in terms of what this means for long-term brain health, we don't know yet, but it'd be really interesting to explore further. And lastly, I'd just like to finish by saying this was more of an observational study. I just wanted to take a small group of participants and see how targeting heel strike force and step frequency, how does it impact these pulse pressure oscillations, in which we found there's a very large oscillation variation between them, and that running and cycling induce different beat-to-beat profiles. And what this means in terms of how does this pulse pressure oscillation impact this shear stress stimulus for improvements in vein vasculature, we don't know yet, but yeah, it'd be really interesting to find out. So thank you.
Nice one, thanks B. Nice talk. Oh, you've already got a question here. So Carolyn has asked, said great talk. Are the oscillations related to the respiratory rate or respiratory changes with exercise modality?
Okay, so Paolettini, he actually looked into this by breath holding during running, and he found that when individuals were breath holding, it did have no influence over these pulse pressure oscillations. So I would say potentially no.
Good answer. You've had that question before. One of the things that one of the things that you see, right, even within those conditions of heel strike, when you're doing the downhill running or the heel striking, the heavy weighted foot, that you see changes within those. The oscillation pattern sort of takes a while to settle and then it changes. So what do you think might be driving that?
So yeah, we did see quite a lot of variation within these conditions, but I believe this was due to changes in the pace, as well as individuals losing concentration, because it's quite hard to try and think how to hill run whilst in the treadmill. But would this be different outside? Potentially, but yeah, so I reckon it's different depending on pace, as well as losing concentration and just being unaccustomed to treadmill running.
Yeah. So what are some of the challenges, I guess, that you face doing that, running downhill on a treadmill? How representative is that of the real world?
There were many challenges. I believe there would be quite a difference to running outside. One, because perhaps there's different in the running biomechanics. You know, you're creating the movement when you're running outside rather than on a treadmill, the treadmill moves for you. So potentially changing the heel to toe running biomechanics, especially during downhill running. Majority of the individuals found it really hard to judge their foot placement, as well as they were anxious, so didn't want to run at their normal running rhythm, as well as a steep gradient, which wouldn't be the case if it was outside.
Cool. A few questions. A couple of them relate to how you quantify the oscillations. One person asking, so Ashley Ackerman asking about frequency-based analysis. Have you thought about that?
Yes, I have considered doing that, particularly fast Fourier transform. But due to this being a master's project and the time restraints, it wasn't included. But I'm looking to do it as I formally write up this research, similar to what Lingira did.
Nice, cool. One more question. So many questions. You might want to jump in and answer these. Did you look at arterial stiffness? Do you think stiffness might affect these oscillations?
So I didn't look into that, but yeah, I definitely think it will affect these oscillations, particularly as well as haemodynamic forces. But yeah, I'd like to look into that actually. I might consider doing some research. Thank you.
That's right. That's from Sandra Willinger, a question, and I guess the consequences or the implications for clinical populations. Nice one. Okay, thanks, B. So we'll move on to our next speaker, which is Sam Weaver, who is going to present his study out of his PhD, which is looking at the protocol-dependent changes. Thank you. Protocol-dependent changes on the cerebral hemodynamics and the neurotrophic responses to exercise. Thanks, Sam. Go for it.
Cool. Yeah, thanks very much for having me, Pat and Caroline. So this is the first project to start my PhD, as Sam said, where we're looking at protocol-dependent changes in hemodynamic and neurotrophic responses, which is part of a bigger project looking at how to assess exercise's impact on the brain's vasculature with in vivo and in vitro techniques. And so we've already covered these topics a couple of times already this evening or this morning. But as we know, cerebrovascular functions improve with exercise, and this is linked to improvements in brain health. And we have this well-established response in continuous exercise peaking at moderate intensities. But much less is understood about higher intensity interval-based strategies. And this is something that we're seeing as research increases. There's now a couple of studies, one by Kirtland and one by Lawrence Lebrecq, that showed that cerebrovascular blood velocity isn't elevated during a single sprint bout, and there seems to be some suppression during that bout. The interesting thing with that being that in terms of cognitive responses, it appears as a reasonably linear response to the intensity, both in continuous and interval-based approaches. There are benefits at higher intensity, and some of those benefits are seen above and beyond what we see at moderate intensity.
Hey Sam, I'll jump in. Can you change your display for us so we can see your presentation? Yeah, you're right. You're right. I know you're not very good with computers, mate. So
yeah.
Cheers, mate. Carry on.
Is that better?
Yep, all good.
So yeah, with this in mind, and with the increased interest in HIIT, and particularly in sprint interval-based protocols with really short intervals, particularly in terms of cardiometabolic health, and the relative improvements in terms of time restraints, we wanted to examine how three very distinct exercise protocols might alter the cerebral hemodynamic and circulating neurotrophic factor responses to exercise. And so to do this, we looked at 3 acute bouts carried out in a randomized crossover order, a moderate intensity continuous exercise protocol or mixed that was 30 minutes of cycling at 65% of VO2 max, a clinical guideline-based high intensity interval training bout or HIIT, which looked at 4 4-minute intervals at 85% of heart rate max, and then a sprint interval protocol of 4 lots of 30 seconds at 200% of watt max, which is somewhat similar to what you see in Wingate, but with a more controlled intensity. Across all of these sessions, we measured MCAV, middle cerebral artery velocity, continuously by transcranial Doppler ultrasound, as well as measuring end-tidal CO2 by breath-by-breath gas analysis, taking 30-second averages, which you can see in the green bars on this schematic, and an area under the curve analysis across the whole of the exercise block. And then we also took blood samples at rest, at the end of exercise, at the end of active recovery and passive recovery. And in the 2 interval-based sessions, we also took an extra blood at this first bout to try and get an idea of what the response difference was between a single bout and a full session of these interval-based protocols. In terms of analysis, the majority of the analysis was carried out by linear mixed effect models with time by condition of fixed effects. And then we used AIC assessments to determine whether or not sex and fitness would be included in the analysis of all the further data. So in terms of the overall response, you'll have already seen this plot in Sam's talk. We saw this clear response in the mixed condition with a continuous elevation over a 30-minute period. And similarly in the HIT, during the 4-minute on intervals, we saw a significant elevation in blood flow, whereas in SIT, we saw this response where we had a slight increase from baseline in the first interval and then very little response over the rest. And when we looked at this in terms of area under the curve analysis, the pattern was as you'd expect. So mixed and HIT were both significantly higher than SIT, and mixed presents significantly higher than the HIT condition, likely largely due to the fact that we've just got more time under the stimulus over a 30-minute period here, as opposed to a total of 16 minutes of intervals in the HIT session. When we look at this in terms of 30-minute blocks of data, we see a significant elevation across the entirety of both the clinical guideline of protocols, so the mixed and HIT protocol, coupled to similar elevations in PET CO2 from baseline. Whereas in the sprint-based exercise, particularly after that first bout, we see a continuous suppression of MCAV, coupled again to a suppression in end-tidal CO2 during these sprint intervals. And this suppression of MCAV was then continued into the active and passive recovery. When we look at the period immediately post-bout, so these recovery points here are the 30 seconds immediately following each interval, we see a very distinct pattern in response in the SIT condition. So we see a significant elevation over baseline during this rebound period that we aren't seeing during the bout itself. And this is in contrast to the HIT, the clinical guideline-based protocol, where we see a gradual decline in MCAV after the bout is finished and the person's drops their intensity back down to 50 watts. Obviously, the potential benefit for this we don't really know, because the stimulus is very short. It's about 15 to 20 seconds of elevation, and we just don't know whether it's the magnitude of that response or whether it's the total displacement of blood over the course of these acute bouts that's driving beneficial adaptations. And as Lebrec showed in their paper, there is an uncoupling of the autoregulatory mechanisms in this single interval rebound, and there's no reason to not assume the same thing isn't happening in these SIT bouts as well. So there is also a possible risk involved with this pattern. In terms of the response in circulating blood markers, we see a very different pattern. So when we look at blood lactate, we see all three exercise stimuli drove an increase in blood lactate by the end of the exercise session. But in the SIT condition, this was markedly elevated above both the mixed and the HIT condition. So by this active recovery point, there was a 5-fold greater increase in blood lactate compared to resting measures in this SIT condition, as opposed to the HIT and the mixed. And not only does this demonstrate the anaerobic nature of the effort, but also highlights the potential benefits of these SIT intervals. As although lactate is a marker of metabolism, it's also a potential signaling molecule in itself. Its ability to bind to the blood-brain barrier and to drive elevations in BDNF and VEGF may have benefits in and of themselves. And then when we look at neurotrophic factor responses, again, we see a significantly different profile to what we see with a haemodynamic response, where in BDNF and VEGF, we see significant elevations in the SIT conditions alone, and these differences are significantly different to any non-significant response we see in either of the other conditions. And then in IGF, we actually see a suppression, but again, it's only in this SIT condition. And in fact, in terms of IGF, this adds to a growingly murky body of water, so to speak, with some authors showing increases during exercise and some showing little or no response at all. And what we are interested in is that we do see this increase as we go into the passive recovery. And it may be that, as this is only 15 minutes after the end of exercise, that this IGF response is delayed, and it may be that we continue to see an increase, but we just don't know. And interestingly, it's this IGF response which shows the least acute benefit. So the responses in BDNF particularly are seen in both acute and chronic cognitive outcome markers, whereas the IGF-1 response doesn't seem to be correlated with the benefits in terms of acute function, but they do appear to be critical in the chronic responses to exercise. And so, as I mentioned, we looked at the AIC information criterion to determine whether any other factors were included in the linear mix modeling. And in fact, only BDNF showed a significant improvement in the effectiveness of the model fit when we incorporated sex. And when we look at sex differences within this data set, we actually see a really interesting pattern of response. So when we look at the SIT condition, that pattern of increased BDNF across exercise, the end of exercise, that's what this X4 point is, and the active and passive recovery is only maintained in the female participants. And within the male participants, we actually only see a slight significant increase at the end of exercise and no significant difference at active and passive recovery compared to baseline. And similarly, in the MIC condition, whilst we saw no response at all in the full cohort, when we split between male and female participants, we see a significant elevation in BDNF at active and passive recovery. Of course, what we don't know is why we don't see any response in this HIT condition. It may be that you either need a very high intensity effort, like you see in this SIT, or a very prolonged exercise stimulus, like in the MICT, and that it's sort of an opposite of a Goldilocks scenario where the HIT session, given it's only 16 minutes of exercise, just isn't enough to stimulate a response, but we just don't know. And similarly, we included female participants in this study regardless of their menstrual cycle phase, so we can't determine what interaction there is between sex hormones and this BDNF measure, just that there is some sort of difference there between male and female. And so in conclusion, we see these very different responses in haemodynamics and circulating factors, and the dichotomy in this response might open the door to us utilizing a more varied strategy in exercise interventions. So Sam mentioned in his talk that we need to look at the whole body, and it may be that we can gain the benefits in cardiometabolic health and exercise adherence with short, hard intervals, and still gain some benefit in the brain through circulating factors. And through incorporating a low intensity exercise, we can then also gain those hemodynamic responses. And so just to acknowledge, thanks to my supervisory team as a whole and all the collaborators we've been working on this project and are working on moving forwards. And thank you for listening if you've got any questions.
Excellent. Nice work, Sam. Really good. I'll maybe start with a question that's probably going to come your way. So how representative do you think these peripheral blood markers are of what's going on in the brain?
So in terms of the benefits you see in cognition, we know a lot more about that than we do about the potential cerebrovascular benefits. And there are issues in terms of the fact that we don't know where they're coming from. Particularly in terms of BDNF, there's a lot of hang up over the fact that it's called brain derived and it may not be entirely from the brain, but there's a decent amount of evidence to suggest that it can come from both, that it can be, that the levels that we see in the circulation, regardless of where they come from, seem to be fairly representative. And there's also a lot of work ongoing at the moment in terms of the acute responses. There's a couple of passive studies that have looked at elevating those factors, and they seem to see cognitive benefits just by elevating factors like BDNF. But yeah, it's a snapshot, and that's why we're working on looking at metabolomics with Rick Dunn at the Phenome Center to try and get an idea of the full profile of responses in those interventions.
Cool. I can ask another question. Why, if there is a sex effect, what do you think's driving that in some of these neurotrophic factors?
I'm not entirely sure. In terms of the responses in moderate intensity exercise, they go against a lot of the data that's been shown in all male cohorts that show no response in moderate intensity exercise acutely. and that the a BDNF response requires a higher intensity. I would imagine there's some interaction with sex hormones. There's no other interaction in terms of the fitness effect, and there's no difference between the male and female participants in terms of age or any other demographic markers. So it does appear to be a sex difference, but we'd need to actually explore that in terms of bringing in female participants at multiple stages in the menstrual cycle and looking at the potential interaction with oral contraception to really know.
Cool. Nice. Here's a question here from Jeremy Walsh. Did you adjust, nice talk, thanks Sam. Did you adjust for plasma volume shifts with exercise and measuring for the neurotrophic factors?
No, we didn't. And it's something that we should look at moving forwards. The relative difference between the 3 exercise intervals may be different. We didn't see any difference in terms of just observationally. There wasn't any particular difference in terms of plasma yield, but it doesn't mean there isn't a difference in the concentration. So it's definitely something that that we'd want to address in future work.
Nice. Here's a good question from Timo. What do you think is the best, given the differences between these protocols, which do you think is the best protocol to use?
So I think in terms of driving a response in terms of just the vascular function, the likelihood is something at a lower intensity, either whether it's continuous or interval based, something at a lower intensity seems to drive the best changes. But I think this is sort of an endorsement of the benefits of a mixed approach. And I think when you look at the success of single protocol interventions and how poor the adherence is to them, I think we do need to just generally move towards allowing people to do any sort of exercise and encouraging them to do as much exercise of whatever type they find enjoyable. And so I think it's a blue sky moment, which is, as Sam will know, very rare in me, that I think it actually just shows that there's potential benefits to all exercise. We just have to find them.
Nice, cool. Okay, I think there's a couple of questions here for you can answer in the chat, I guess. But let's move on. Thanks, Sam. Great stuff. Leena Shoemaker is our final speaker. As I mentioned, she did a PhD out of New Zealand with Jim, and I was involved remotely. And this is one of her studies that she did through the PhD. So some really neat methods used here that I know she's looking forward to sharing. So thanks, Leena. Go for it.
Am I still muted?
You're all good. Go for it.
Awesome. Okay, good. My video disappeared and somewhere into the Zoom world, so wanted to confirm. Well, thanks, guys, and thanks for sticking around, everyone who's still here. Some really interesting stuff going on in the cerebrovascular world in this seminar series. It's been a really neat opportunity to see everybody's work and really come together in this interesting time of our lives. So let me make this big and swap presenter. So as Sam introduced, I'm going to be presenting some of my swimming data that I collected during my PhD, which was, excuse me, completed at the University of Otago in beautiful Dunedin, New Zealand. Here's the beach in Dunedin. And I don't need to convince this audience that exercise is good for us. It's good for our mental health, longevity, acutely and chronically. It can improve cognitive function. And we know that it's a benefit to vascular health. But when we talk about swimming specifically, we're thinking about quite an accessible mode of exercise. This is accessible to people with disabilities, with injuries, particularly because it's very low impact. And as Sam so greatly showed his data earlier that ties into what I'm going to be presenting, we have found data that upright leg-based aquatic exercise augments the cerebral blood flow response to an intensity-matched land-based exercise. And in combination with some swimming-related factors that I'll present in a moment, we see that swimming really is a unique stimulus, or aerobic exercise in the water is a unique stimulus for cerebral blood flow regulation. Now, it's definitely an integrated response when we talk about cerebral blood flow regulation, but water immersion itself has numerous pronounced cardiorespiratory effects that directly influence cerebral perfusion control, some of which include carbon dioxide, blood pressure, and cardiac output. And when we talk about swimming, particularly against upright exercise on land, we first think about the posture differences. So now we're in a prone posture compared to upright posture in most sports on land. We also consider that we're in a new environment. We're in water immersion, so we have effects of hydrostatic pressure. And we have some breathing changes. So there's momentary apneas, but we also have some CO2 retention just from being in the water, as well as it being an upper body movement mostly compared to a lot of land-based exercise, which is a lot of lower body. So this presents a unique stimulus for cerebral regulation, but the cerebrovascular effects have not really been examined in either acute or chronic contexts. And more so, the separate and combined effects of these factors on cerebral perfusion have not been examined. So that's what we sought out to do. Our primary aim was to determine the cerebrovascular response to moderate intensity swimming and identify the independent and combined effects of posture, water immersion, CO2 retention, and motor involvement. Now, I have a particular interest in cognition, and so we added a secondary aim to examine the acute effects of swimming and changes in cerebral blood velocity on different aspects of cognition. We got 2 groups in. Each group were highly trained athletes in either swimming or land-based exercise, most of which were runners. And they were matched for BMI, age, and VO2 max. So when they came in on the first day, we did a familiarization, and this was including a VO2 max test on the treadmill. And then after sufficient rest, we brought them in to do a swimming heart rate max test. And this is in the swimming flume that's at the School of Physical Education, Sports and Exercise Science at the University of Otago. And this is a really cool piece of kit. I could do a whole other talk on just everything that this offers. But essentially, we call it a water treadmill. And it's different than the water treadmill Sam was talking about earlier, because we change the pace or the speed that the water is coming towards the participant. So if you can imagine, this participant tries to stay in place here as we change the speed that the water is coming at them. So this offers a great environment to look at a participant with all of our integrative physiology variables while they're swimming or doing exercise. So during this swimming heart rate max test, we were looking at end tidal CO2, we were measuring heart rate to calculate heart rate reserve, and we were measuring RPE. And this was important because we wanted to define what each individual's submaximal or 50% heart rate reserve would be and what the corresponding end tidal CO2 was for that person. Then they did cognitive test practice, and they practiced enough times to kind of diminish the worry that learning effects would come into play in the next session. During the experimental visit, we wanted to do a swimming delineation. So as you can see down here with all of my little stick men, we've got the effects of postures. So we have upright and prone posture on land as well as in water. And then we added CO2 using a CO2 clamping system to match their personal end tidal CO2 change during submaximal exercise. And again, this was in both postures on land and in water. And then we got them to do 20 minutes of breaststroke swimming at 50% heart rate range. For this entire duration, we measured cerebral blood velocity in the MCA, which you've heard before. This is using transcranial Doppler. We have a fantastic model here, my favorite photo to use. And we also measured end tidal CO2 and blood pressure. The cognitive test that we used was developed at the University of Otago, and it's similar to the Stroop test in that it's a reaction time test measuring executive function. So it's 3 different tasks included in one battery. The first task is called Pro, and the Pro is green squares appearing on the screen, and the participant must press the same side button that the green box appears as fast as possible without sacrificing accuracy. Then you get a group of red boxes appearing, and the rule changes for the red boxes. Now you have to press the opposite side button as fast as possible without sacrificing accuracy. And Pro-Ante is the last task of the battery, and that's a combination of green and red boxes, which is definitely the hardest version of the battery. We adjusted for any changes in error rate, and I will be reporting an accuracy adjusted reaction time, ART. So this was measured on land in an upright posture, in water in an upright posture, and then after the swimming. Here's a picture of our participant in the flume doing the cognitive task, just the setup that we had here. Now, I will say that I'm going to be talking a little bit more about the methodology because it was a lot of fun, and I think the pictures are kind of cool showing what we can do even down in New Zealand. This is our participant in a prone posture on land, and I have the MCA trace on the left side. So what you can see, we have the TCD setup, we have the CO2 clamping system, and then we have the Omron BP that we used on land and in the water. Prone posture in the water, as you can see, we use facilitation aids to help, or flotation aids to facilitate a passive float. So they weren't using, minimizing the effort involved in staying in the water. We also had the water running so that it was very slowly, so it was keeping them in that posture. Then we added CO2 while they were in the water. As you can see here, we moved the box to match them. And then we did the breaststroke swimming for 20 minutes. So this is a video. I think it's kind of funny. We went to great efforts to try to get as many variables as we could. And the video is of me trying to measure blood pressure with somebody swimming. So this is a national champion swimmer doing a fantastic job of one arm stroked while I was using feeling, because I couldn't hear, to measure blood pressure. And if anyone's interested, it was around 188 over 64. We didn't report this in the study, but just a fun little video to see how somebody's actually swimming in the flume. So the results, as you can see, we've got on the x-axis, we've got all the different conditions that the participants went through, and on the y-axis, we've got MCA, so middle cerebral artery velocity. And this was the mean. Now, the first thing I want to point out is that upright posture compared to, so swimming compared to upright posture increased cerebral blood flow by about 20%. And the second thing I want to point out is that in all conditions, it appeared like there was a main effect, for land-based athletes, such that swimmers had 17% lower resting MCAV, despite the condition. Now, to address the main question of the effects of posture, water immersion, end-tidal CO2, and swimming, we did multiple ANOVA comparisons. So looking at the effect of posture, we did upright versus prone posture on land, and as expected, MCAV increased by about 11%. Water immersion in a prone posture did not statistically change MCAV. Now, I don't show this here, but it is important to note that upright posture on land in the water, it was higher in the water than on land, but we don't see that effect when we look at prone posture. Next up, end-tidal CO2. So now we have prone posture in the water against prone posture in the water adding CO2. And you can see here that MCAV was increased by about 14%. And when we look at swimming, there was no statistical difference when we just add the motor involvement compared to a matched CO2, water, and prone posture immersion. So now onto the cognitive results. We've got pro at the top here, we've got anti, and then at the bottom we've got the pro, anti, and I'm showing you their scores on land and the scores in the water. And there's no significant difference here. So water immersion did not improve cognition. And you might be thinking, why would I think that water immersion would change cognition? Well, water immersion in an upright posture is associated with increased cerebral blood flow. And so part of my thesis was addressing if I could acutely change cerebral blood flow, does that also result in cognitive changes? And I plotted it so we could tell that story a little bit more clearly. We have percent change MCAV on the x-axis and percent change pro-ART, so this is the visuomotor, the green boxes only, plotted against. And you can see there's no real correlation happening here. So we can conclude that in this study, an increase in MCAV acutely did not result in improved cognition. Now for the second question, we wanted to know if 20 minutes of swimming could benefit cognitive function. And the only difference we found was in the pro-visuomotor response time, where swimming improved cognitive performance by 3%. And again, the same plot, except this time we're looking at pre versus post-swim changes for MCAV against the pro-ART. And even without this outlier here, we didn't see any significant relation between these variables. So the improvement in cognition was likely not due to an increase in MCAV. So in summary, swimmers may have lower resting MCAV mean. Posture and end-tidal CO2 seem to have the largest impact on the change in MCAV during swimming. And 20 minutes of breaststroke swimming did improve cognition, albeit in limited domains. So for our future directions, we were really thinking about whether swimmers have these vascular adaptations, and particularly with our main effect that swimmers had lower cerebral blood flow velocity. So we brought back a subset of participants and we measured ICA diameter and flow, and we did not find any difference between the groups. So the jury's still out on that one. Could have been a sampling bias, or we have some other theories. Then how can swimming benefit the brain health of older adults? Sam touched on this a little bit. However, in the context of this study, we're really thinking about prone posture swimming. Breaststroke is quite feasible for older adults as well, albeit a little bit more technical than walking. But it's definitely an avenue to consider, particularly in relation to the cognitive benefit of exercise training. And with that, I'd just like to acknowledge the supervisory committee, including Sam Lucas all the way in the UK, and the funding support that came from the School of Physical Education, Sports and Exercise Sciences from the University of Otago. And thank you very much.
Nice one, well done, Lena. Really cool, interesting study. Yeah, sorry, I made you put that video in because I think it's so brilliant watching what is a real classic Otago approach to doing science. So,
Huey ingenuity, Sam. It's a real thing. I tried my best.
That's good. So I guess a question that that I have from these data really is, I guess you touched on it in your talk, is why do you think swimmers have this lower blood flow, and what do you think the functional consequences of that might be? Is it all the chlorine?
Yeah, I don't know, to be honest with you. If I'm being very honest, I would like to do the study again and get a totally different sample of swimmers and a totally different sample of runners and see if that effect stays. I don't know if it's a sampling bias. I don't know if something else is going on in the brain. It could be. I mean, a lot of your work that you're talking about is this chronic adaptation with a higher stimulus. So I'm open to ideas about what's happening here. I don't know that lower velocity is functionally bad if we're in a young, healthy group. I think long term, we're thinking that higher velocity is preventative against some cognitive deficits, but I don't know.
Yeah, and I guess and even velocity or blood flow, or we're talking in the context of global our flow measure.
Exactly.
So Pat asked the question here. So we've talked about a whole bunch of different exercise modalities now. So do you think swimming is better than walking or running in water? So if we're going to use water, we're better to lie down and do it, or should we walk or run? Or, I mean, we can also cycle in water. There are aquatic bikes as well. So which one of these do you think might be best? Which I guess boils down to prone versus upright, yeah?
Right, yeah. Okay, my big thing with exercise is, are you gonna do it, first of all? So if I find swimming in a prone posture a lot more enjoyable than walking, but I know a lot of my friends prefer upright. So first of all, let's just address the fact that with the preference and excuse that. One thing I would like to look at, or we should have maybe done, is look at cerebral blood flow during swimming compared to an intensity matched upright exercise in the water. I have a feeling that it's going to be higher cerebral blood flow if that's what we're using as the most stimulus and the best reason to choose the exercise. Then maybe the upright water walking would give more stimulus for adaptation compared to prone posture. You just don't have that gravity effect when you're in the prone. Different hydrostatic effects, and
Cool. So a couple of questions here. If I can keep up with the screen. So Kurt Smith, expiratory resistance. How do you think that might impact on the cerebral blood flow response?
Probably. I haven't thought about that much. Yeah, I'll get back to you.
In terms of dead space with the clamping system, did you have any of that in terms of, is there extra dead space and CO2 retention just from that, how does the clamping system deal with that?
Right. So there is a methodological comment that I probably need to make to answer that. They weren't, so they wore the snorkel, which would have some dead space in it during the familiarization session where they did the heart rate max, but they didn't wear it while they did the 20-minute swim. So the final cerebral blood flow during swimming measure that I had didn't include the snorkel. And although we didn't see much difference, you know, at rest when they had it on versus when they didn't, I can't exclude the possibility that that had some effect. So the other thing is that they weren't doing momentary apneas as well, because at least when we were measuring CO2, they had the snorkel. So they didn't need to hold their breath. And most of the time we told our participants when they were swimming to hold head out swimming. So they weren't holding their breath, and we really didn't collect data to look at that question.
Yeah, it's interesting, yeah, different modalities. And rowing's the same, your breathing is very linked to the movement. And yeah, I guess you're changing it, you know, with what you typically do with swimming. So yeah, cool. Let's have one more question.
Thanks, Gary.
Temperature, water temperature. What was the temperature the participants were exposed to in the water, and could this make a difference?
Okay.
I guess particularly if we're talking cooler water and full face immersion.
Yes, for sure. We did a lot of pilot work to look at the temperature of the water. It was 28 to 30 degrees Celsius, and I calculated it earlier to be 82 to 86 degrees Fahrenheit. So that was something that we needed to be careful of, because I do think temperature, if they were too hot or too cold, even during those conditions where they weren't moving, if they were in the water too long without moving, they still would get cold at 28 to 30 degrees. So we tried to go as fast as possible, and did a lot of pilot work to make sure that we would minimize the effective temperature on our control. And that was also why we didn't randomize the in water versus the out of water conditions, because going in the water to out, being wet would just make you colder for the other conditions. So when they were in, they were in.
Nice, excellent. Okay, well, thank you, Lena, and thank you everyone for staying in and listening. It's now 10 o'clock at night. I think I'm probably the main culprit. I think I spoke for too long, perhaps. But thank you for listening. Thank you for the questions, and I'll throw it back to Carolyn.
Thanks so much, Sam. I'm just going to advertise for the next session as well. So from one Shoemaker to another, we have Dr. Kevin Schumacher will be chairing the next session on August 17th. And so he'll be chairing and be the keynote speaker as well. So look forward to Dr. Shoemaker's session on August 17th. So thank you everyone for attending again today. I'll be posting the video over the next couple of days, and also sending out this flyer for registration for the next session on August 17th. So thanks so much.